afternoon for my online presentation of my dissertation defense. Very briefly, I want to go through the topics that I'll be covering in the next little less than an hour. I'll be starting with an overview of chloride in freshwater ecosystems. So this is where it comes from and why it is a problem. Next, I'll describe my laboratory study of the toxicity of chloride with a focus on the importance of running tests under environmentally relevant conditions. Then I will discuss a study where I examined the biodiversity of aquatic macroinvertebrates in streams and their association with chloride concentrations in those locations. And finally, I'll describe my study of a best management practice that is intended to limit the amount of chloride that reaches the stream ecosystem. So now I'll begin with an overview of chloride in freshwater streams. The use of road salt to improve the safety of travel following winter snow and ice storms is a relatively recent phenomenon, first gaining traction in the 1940s. Since that time, this practice has gradually spread to include most parts of the world where snow falls. The use of salt in the United States now exceeds 20 million tons per winter, and the purchase price for cities to buy that salt in bulk is about $50 per ton. And so about a $1 billion annual investment is made in safe transportation. But there are several negative consequences to using road salt. First, salt is corrosive to concrete and to metal. In a 1992 study by Vitaliano, they estimated that 1 ton of salt, which, as I mentioned, costs about $50, will do about $600 in damage to infrastructure. So that's just the roads and bridges. That does not include the damage to people's vehicles. And so therefore about a $12 billion damage bill just for roads and bridges across the United States every year. So the salt that's on the road dissolves, and that salty water may travel from impervious paved surfaces like sidewalks, parking lots, and roads into areas where there's vegetation. Salt at these concentrated levels is toxic to many plants, including grass and street trees. This is also a concern in places where plant-based stormwater solutions like rain gardens have been implemented, because this is also toxic to most of those plants. And so that salty stormwater runs off the pavements, into the grass, and then also into storm drains. And from those storm drains, it's transported into as runoff into nearby streams. So these are freshwater streams, and the animals that live in these streams and rely on them for survival have adapted to freshwater conditions. Yet every winter, aquatic life is exposed to highly concentrated salt from runoff, potentially creating toxic conditions for them. But salt doesn't just stop in the streams. It can migrate into groundwater, and it can accumulate in lakes. And so both surface water and groundwater serve as drinking water supplies. The increase in salt in these drinking water supplies raises concerns about water potability, especially for those who have underlying health conditions like heart disease. Now, before we move on into the work that I've been doing, I wanted to give you a few relevant numbers to keep in mind. Natural chloride concentrations for streams in much of the United States are less than 40 milligrams per liter. There are definitely exceptions to this, and those are generally based on the geology of the area. Every state sets their own water quality standards for different water uses, and then those are approved by the US EPA. In Missouri, there's a standard for drinking water, and that includes a limit for chloride of 250 milligrams per liter. This happens to be similar to the chronic aquatic life threshold, which is set at 230 milligrams per liter. And then there's an acute toxicity threshold of 860 milligrams per liter. And when speaking about acute and chronic exposure levels, when talking about water quality and standards, those are generally in terms of about a 1 hour exposure for acute toxicity and a 4-day exposure for chronic toxicity. And then just as a point of reference for chloride on the high end, seawater contains about 20,000 milligrams of chloride per liter. Okay, so let's move on now to my laboratory study of chloride toxicity. This work is focused on developing a better understanding of the toxicity of chloride under winter conditions. Traditional toxicity assays are conducted under highly controlled and standardized conditions with regard to features like lighting, temperature, media, and feeding. For example, the temperatures that are used are generally near room temperature, so this is about 20 degrees Celsius. The standardization of testing is important because it improves the comparability of studies being done in different labs or different test subjects. But the environmental conditions when road salt is being used are much colder than this. Since physiological responses of organisms are often impacted by temperature, it is possible that traditional assays that have been used to develop the water quality standards that were shown a couple of slides ago may not actually be representative of the responses of organisms under these colder winter temperatures. So my first study tests the hypothesis that both lethal and sublethal responses to road salt can differ based on the temperature of the water. To test this hypothesis, I collected individuals of the crustacean Gomeris mitis from Kiefer Creek and the mayfly Stenonema femeratum from Sugar Creek. Members of these genera are often used in toxicity assays, and these particular species were selected due to their local abundance and their relatively broad distribution. Both living in relatively large parts of the eastern United States, with the Stenonema femeratum extending into Canada. So these two figures in the middle show temperature and chloride in the two source streams between November of 2017 and June of 2019. Chloride concentrations, shown in black, are at or below 100 milligrams per liter for much of the summer, but do have some spikes in the winter as a result of de-icing with road salt. Kiefer Creek has a much smaller set of chloride spikes, and that's partly due to the fact that the watershed is mostly forested, and that's demonstrated by the green. Meanwhile, Sugar Creek has higher spikes, and that watershed has a lot more urban development, as shown with gray in that map. So the water temperatures are shown with the brown lines, and we can see that summer temperatures are around 20 degrees Celsius, but winter temperatures are generally below 10 degrees, and in Sugar Creek even reaching 0. Temperatures in Kiefer Creek, you may notice, have a slightly narrower range, and this is due to the large number of springs which feed into the stream and moderate the water temperatures. All right, so in my study, I tested these two species for 7 days under both cold and warm temperatures. Temperatures were moderated slightly for Gomeris to reflect the conditions in the spring-fed source streams. Test concentrations of chloride were based on the results of a preliminary assay to kind of make sure I had a good range there. The control concentration of the source water, or the control concentration, excuse me, is the source water collected from the stream with no salt added to it. But notice that the Stenonema control concentration is already higher than that chronic toxicity threshold of 230 milligrams per liter. For each of the treatments, 40 total individuals were tested in 4 chambers of each containing 10 individuals. However, when the individuals were identified to species following the completion of the experiment, it turned out that several of the mayflies belonged to a different species, and so that's why a couple of the treatments had fewer than 40 individuals. And finally, for the Gomeris only, each test chamber received a pre-weighed silver maple leaf, and that leaf was re-weighed after the experiment was completed as a non-lethal test of chloride's effect on feeding. So I'm going to show you the results of the lethal toxicity trial here in a moment. But first, I wanted you to note that the 2-day and 7-day LC50 values represent the concentrations of chloride that would be expected to kill half of the population after 2 or 7 days. And so when we see numbers here, the higher concentrations indicate a greater tolerance for salt. These results were generated using a probit analysis and were evaluated for significance based on the overlap of the 90% confidence interval. So after 2 days, there was a difference between the cold and warm treatment for Gomeris mitis, but it was not significant. And then for the Stenonema, over half of the animals in all treatment levels survived for at least 2 days. And so the LC50 is higher than the highest test concentration, which was 14,500. So this high tolerance may be related to conditions in Sugar Creek, where anthropogenic land uses are already contributing salt to the stream. And so that may have resulted in some sort of adaptation or plasticity in the population, but I can't be sure on that. So after 7 days, both species did exhibit a significantly greater tolerance of chloride in the cold temperature when compared to the warm temperature. The LC50s in the warm temperatures are in line with what tends to be found in the literature for these genera. In the non-lethal portion of the study, I measured the amount of leafy material that was consumed during the 7 days of the trial. From these images, you can see some typical examples of what was eaten at relatively low salt concentration. Here you can see that much of the leafy material is gone from this leaf, but then at the higher concentration, the leaf is almost completely intact, except maybe a little bit eaten there in the center of that circle. Okay. So in this box plot, the cold treatment is represented by blue, the warm treatment represented by orange. Any boxes that share a letter are not significantly different from each other. So for example, there were no significant differences in the two lowest concentrations, and that was regardless of temperature. Now, once we get to 1400 milligrams per liter, the amount of leaf consumed was still the same within the temperature grouping as the two concentrations below, but there was a significant difference with the animals in the cold treatment consuming less leaf mass than those in the warm. And then this carries through at the higher concentrations chloride treatments as well. Now, looking more specifically at the chloride concentrations, it's apparent that somewhere between 1400 and 3600, there is a strong decrease in feeding. And after this, the warm treatments remain steady, while the feeding then ceases entirely in the cold treatment at the highest chloride concentration. Now, if you recall from the lethal toxicity study, the LC50 in the warm treatment was 2,700, which coincides with this drop in feeding. But at a chloride concentration of 3,600, despite there being more than twice as many animals still alive in the cold treatment, there was less feeding in the cold treatment. So let me repeat that. Twice as many animals alive, but they ate half as much food. So to recap this first study, my hypothesis was that lethal and sublethal temperature or responses to road salt would differ based on water temperatures. Regarding lethal toxicity, aquatic macroinvertebrates are more tolerant of chloride concentrations under colder temperatures, as would be found in streams during the winter. So this is good because it suggests that the toxicity thresholds that were developed based on assays conducted under warm conditions would also be protective of invertebrates from lethal conditions during winter temperatures. On the other hand, the feeding response of invertebrates under cold temperatures suggests that lethal impacts or sublethal impacts could be very important. The observed reduction in feeding has strong implications for an array of sublethal impacts. At the population level, we may expect to see population declines due to repercussions in body weight, which changes brood size, time to maturity, and other responses. We can also expect changes to ecosystem functioning. For example, gamerids and other amphipods like the Gamaris in our study are important in the processing of leaf litter. They shred and consume the leaves that fall into streams, both feeding themselves and also providing smaller particles that are then eaten by other invertebrates. So as their winter feeding slows and population sizes decline, nutrient cycling would also decline. So this is what I found when looking at small numbers of invertebrates at a laboratory scale. But now we're going to move into my second study, which explores the response of aquatic macroinvertebrate assemblages in streams used with different levels of chloride inputs. So we know that chloride above certain concentrations will impact freshwater biological communities. Many prior field studies of the effects of chloride on in-stream assemblages have characterized the level of chloride using just a few grab samples, providing instantaneous snapshots that are unlikely to catch chloride at its peak. So what are the peak concentrations? And what are the specific drivers that are influencing how invertebrate assemblages are responding to increased chloride? Is it purely a matter of high concentrations being bad, or does the duration of the exposure play a role as well? These questions cannot be answered using such limited chloride grab sample data. So for the second study, I used data loggers to establish a chloride record with high temporal resolution in order to explore two hypotheses on the effects of chloride on aquatic macroinvertebrate assemblages, and those are tested using different levels of taxonomic identification. So my first hypothesis is that taxa richness will be more strongly correlated with urban development than it will with chloride. Urban development has long been identified as a driver of diversity loss in stream ecosystems. And so this hypothesis reflects the thought that as the broadest taxonomic level, or at the broadest taxonomic level, the different effects of human land uses will wash out any more specific effects from chloride. My second hypothesis is that Ephemeroptera diversity will decrease as chloride concentrations increase, reflecting the sensitivity of this order to chloride. So this project began with the help of 36 trained volunteers with the Missouri Stream Team program who monitored chloride in the winter at 63 sites across St. Louis County, Missouri. They've taken over 2,800 chloride grab samples since 2012. And on this map, we can see a summary of the maximum chloride concentrations that they found in sites across the county. So this area is the city of St. Louis, and then there's decreasing urbanization as we move out toward the west. The sites in blue are sites that remained below the chronic toxicity level. The sites in light blue stayed below the acute toxicity level as the maximum concentration. And then sites in yellow, orange, and red all had concentrations that were over the acute toxicity threshold to 1 degree or another. And then the size of the dots are related to the size of the watershed that drains to that sampling. So we can see that as we move away from downtown, there are fewer sites with high chloride concentrations. Now, I want to remind you that these are grab sample data, kind of like I talked about before, that are giving snapshots of a point in time. And so these represent the maximum value in some cases of only 5 samples, and others represent the maximum of over 100 samples. So I always like to remind people that we have to take this data with a grain of salt. And normally I would be in a room with a bunch of people and I would be getting groans and giggles and laughs and stuff, and waking up whoever might have been falling asleep. So hopefully that's been done and we will move forward. Okay, based on volunteer data from the previous slide, I selected 15 streams that showed a range of maximum chloride concentrations as well as a range in the percent of winter samples that were above the acute and chronic toxicity threshold. So these are the sites and their watersheds. The background color in the city area indicates the level of urban development, with high density development being in darker gray, less dense development in lighter gray, and then non-urban land uses shown in white. 3 samples of aquatic macroinvertebrates were collected during each of 3 field visits during the spring and fall of 2017 and the spring of 2018. Invertebrates were sorted and placed in taxonomic groups under 8x magnification. And so the first macroinvertebrate metric for this study was the richness of invertebrates that was identified at this level. Members of the order Ephemeroptera, or mayflies, were then further identified to the level of genus and then counted. Shannon diversity was then calculated as the second biological metric. Conductivity was recorded at 15-minute intervals over the winters of 2017 to 2018 and 2018 to 2019. There is a linear relationship between conductivity and chloride, and so this allowed the generation of a chloride data set covering 316 days over the 2 winters and into the springs. These data were then summarized into 6 chloride metrics that I'll talk more about in just a minute. And in order to identify non-chloride factors that might also impact the invertebrate assemblages at these sites, I made measurement of several habitat metrics and landscape level factors. The 5 metrics I included of in-stream habitat were percent canopy cover, percent stream bed covered by algae, percent of the bed composition as sand plus silt, percent of the bed as cobble, and then the percent embeddedness of the substrate, so how much sediment is coming in on top of that sediment. 3 landscape metrics of watershed conditions were also used, and those were watershed area, percent impervious surface, which is used as a surrogate for urban development, and then the percent of roadway coverage. So these box plots summarize the 15-minute chloride data over the study period. The thin lines represent the full range of the collected data, so maximum and minimum. The blue boxes represent the interquartile range, or the middle half of the data. And then the horizontal bar inside of each blue box represents the median concentration. So half of the collected data were above this and half were below. And note that several sites have maximum concentrations that are approaching 8,000. And one site even had a maximum concentration of over 25,000, which puts it well above what you would find in the ocean. So all of the sites reach above the chronic toxicity threshold, shown here, but 3 sites stayed below the acute toxicity threshold, shown here as this red line. So given all of this data, what is the best measure of chloride in this situation? So rather than select a single measure, I included 6 different metrics of chloride: the maximum concentration, the median concentration, the total duration or amount of time that the concentration was higher than 230, or the chronic threshold, over the study period, the total duration when the concentration was above the acute toxicity threshold, and then also the maximum continuous durations, so the longest single period of time, the duration of that, when the concentration was above either the acute or the chronic toxicity levels. So I ran a Spearman rank test, which identified strong correlations among the 6 metrics. So a principal component analysis, or PCA, was used to reduce the number of correlated variables. The first 2 axes of the PCA accounted for nearly 90% of the variance in the chloride metrics. The first PC axis predominantly identifies locations with high or low chloride concentrations. The second PC axis differentiates between locations in which the chloride concentrations tend to have a higher maximum concentration and more frequent concentrations above the acute standard, versus those in which the concentrations have a greater tendency to exceed the chronic standard of 230 milligrams per liter, and the median concentration is higher. And so to help understand what this looks like when you're looking at the whole data set, I've included the time series of chloride concentrations from Monsanto Sunswept Creek at the top and the River des Peres at the bottom. The solid red lines represent the chronic toxicity threshold, and the dashed red lines represent a concentration of 6,500 milligrams per liter. And that was selected just because that's where the 2 peaks show up here for the Monsanto Sunswept Creek site. And then the blue bars represent periods of 1 month or longer when no chloride concentrations were higher than that chronic toxicity threshold. So let's first consider the maximum concentrations in these time series. The chloride concentrations at the highest peaks in Monsanto Sunswept Creek, as I said, are right here around 6,500. But if we look at the River des Peres, there were several peaks at that level, but then we have 3 peaks that are quite a bit higher than that. Again, one of them over 25,000, another over 18,000, and a third that's over 10,000. So also in this first winter, the concentration in River des Peres spent quite a bit of time in this region above the acute toxicity threshold. And so that matches up with these lines on the PC axis. All right, and so while the River des Peres has some tremendous winter concentrations, it also has much longer time spans, shown here in blue, when the concentrations stay below the toxicity threshold. But not just the periods in blue, but also in this area here and in the second winter as well. Now, if you compare that to what we see in Monsanto Sunswept Creek, the second winter, once it went above the chronic toxicity level, it stayed above that level for the entire measured part of the winter. And then the blue area where it stayed under the threshold was also much more brief than in river de pear. And so you end up with a higher median and a longer duration above 230 milligrams per liter. All right, so aquatic invertebrates, as I mentioned, were collected in the 15 streams of our study area, and then those were sorted into 31 taxonomic groups. These images show representatives of 9 of those groups. The presence or absence of each group was determined based on aggregating all 9 samples collected at each site. And then the richness that was found in each site ranged from 12 to 26 taxa. So this is just a listing of the various taxa that were identified from samples in the study. Classes are shown in bold, orders are shown in dark unbolded text, and families are listed in blue. And identification, as I mentioned, was done under 8x magnification. So some of these groups were present in nearly every sample, specifically aquatic worms and midgefly larvae. Other groups, including riffle beetles, sow bugs, springtails, and flatworms, were found in at least one sample at most of the sites, but with a range of frequencies of whether it was in just one sample or all the way up to 9. Among the rarest groups were the crayfish, cladocerans, stoneflies, and several of the other types of beetle larvae, which were found at very few sites and very infrequently within the samples. Looking more specifically at the diversity of the Ephemeroptera or mayflies, I identified individuals belonging to 7 genera in 3 families. Notably, of the more than 7,000 individuals identified, over 96% of them belonged to the genus Baetis. The Shannon-Wiener diversity values for Ephemeroptera range from 0 to 1.12. Predictive models were generated for both of the both taxonomic richness and mayfly diversity using the 10 environmental variables. This was accomplished using model selection based on forward stepwise regression. Model comparisons were made using AIC scores corrected for small sample size. So the model with the lowest score is viewed as the most reasonable predictor. This approach maximized model fit while maintaining the number while minimizing the number of explanatory variables that were included from the list of environmental metrics. So the final models for the 2 aquatic invertebrate measures are shown here. Looking first at taxa richness, we see a positive relationship with both watershed area and with the second principal component axis. The increase in taxa richness with increased watershed area follows the predictions that are made for small streams in the river continuum concept, which basically states that further downstream, there's often a greater heterogeneity of habitat and food resources available, and so that can support additional taxa. Meanwhile, the influence of the second principal component axis suggests that the type of exposure to increased chloride concentrations is important to aquatic macroinvertebrate assemblages. So the positive relationship between the axis and taxa richness indicates that sites with higher median chloride concentrations or a longer duration of exposure to the chronic toxicity threshold may maintain a greater number of species than sites that have greater maximum chloride exposures or longer acute exposures. So this suggests that events categorized as acutely toxic may have a greater impact on taxa richness than long-term low-level toxic exposures. So moving on to the diversity of the mayflies, we see a negative relationship with the first principal component axis and with the percent algae cover. PC1, as I mentioned before, is primarily a composite expression of chloride concentration with all 6 chloride metrics positively related with the axis. Then the negative relationship of mayfly diversity with the axis suggests that increased chloride overall has a negative impact on mayflies. There's also a significant negative relationship with the percent algae cover. The genera of mayflies that were found in our study all feed as collector-gatherers or scrapers, meaning that algae will be a dominant component of their diet, so this may contribute to that relationship. Right, so to recap my second study, my hypotheses were first that taxa richness would be strongly correlated with urban development rather than chloride, and second, that mayfly diversity would decrease as chloride concentrations increase. My first hypothesis was not supported. Not only was chloride exposure a driver of taxa richness, but the amount of impervious surface, which was used as a surrogate for urban development, was not included in the best model. So what's especially interesting about the relationship with chloride is that the PC axis for concentration of chloride was not important. So just the value being high or low was not the important part, but rather the type of exposure, which incorporates the duration and the magnitude of exposure. To me, this finding is highly important because it demonstrates that there are aspects to pollution exposure that we've been missing in a lot of our approaches to evaluating water quality and to how we've been studying how chloride impacts aquatic invertebrates. My second hypothesis was supported by the model. Increased concentrations were in fact connected with decreased mayfly diversity. This relationship existed despite the generally depauperate mayfly assemblage in these streams. Only 5 sites supported more than 2 genera of mayfly, and the maximum one site had 5 genera. In addition, the majority of these genera are considered facultative or tolerant of pollution, and more sensitive species that are known to exist in neighboring counties were not present in these samples, making it all the more interesting that the remaining types of mayflies would in fact have a measurable negative relationship with chloride. All right, so now we're going to change gears for the third study of my dissertation. I've been talking about chloride in the context of its impact on aquatic invertebrates, but having knowledge of a problem and its consequences does not often provide solutions. So here I'm going to describe a study of a best management practice that is intended to limit the amount of chloride that reaches stream ecosystems. Salt accumulation in streams is a problem that needs to be solved via resource management and stormwater management. Much stormwater management is done using solutions like rain gardens and stormwater detention basins. And these are designed to allow stormwater to filter through the soil or a soil-like medium, and then the water can enter the groundwater or stormwater system. Many pollutants like nutrients and sediments are effectively removed in this process. But when we're talking about road salt, much of the salt remains dissolved and passes through the filters. So the only truly effective way to decrease the winter salt load to streams will be to decrease the amount of salt that we put on the roads in the winter. So I wanted to study a method that some cities and other municipalities are using to cut their salt use, and that's the application of salt brine. So my hypothesis for this final study is that cities that use brine as a best management practice, or BMP, will contribute less chloride to stormwater systems and thereby into streams than cities without brine. So what is brine? Brine is salt water, and so the practice of brining involves taking the same solid rock salt that's normally spread on the roads and dissolving it in water to create a 23% solution and spraying it on the road before the storm. So there are several suggested benefits of brining. First, it can be done up to 2 days before a forecasted storm, which allows road crews to work during regular business hours and cuts down on overtime costs for cities. According to various studies conducted on highways, the same road clearing effect can be achieved with brining using half or less the amount of salt. And so this reduces a lot of the negative consequences that I mentioned in the introduction. Now, there are also some potential drawbacks to the use of brine, particularly in areas like St. Louis, where it's often difficult to predict the form that a winter storm may take. Often a storm might begin as rainfall and then transition into snow or ice. Under these circumstances, if brine has been applied, the rain will wash it from the pavement and road crews would need to return and spread the rock salt as well. And this would end up actually increasing the load of salt rather than decreasing it. So several prior studies of the effectiveness of brining have been conducted on highways and on parking lots, but none have included real-world applications of brine for storm in residential areas or the use of the best management practice by multiple municipalities. So for this project, I studied 3 pairs of cities. Each pair was selected to include one city that has a brining system, shown in dark blue on the map, and one city that does not have a brining system, and those are the ones in light blue. Pairs were set up as neighboring cities in order to help control as much as possible for the variability of storms as they cross through the region. Pairs were also selected to try to minimize social and economic differences between cities. Socioeconomic factors like city revenue, per capita income, and the density of urban development might impact salt use within city limits due to things like the city's salt budget, resident expectations for road conditions, and the likelihood of residents to use salt on private driveways and sidewalks. So in order to confirm the socioeconomic similarity of the neighboring paired cities, a principal component analysis was run on 13 demographic, economic, and land use metrics. This PCA includes the 6 study cities, but also added 3 non-study cities of comparable size in St. Louis County as an outgroup. The first 2 PC axes accounted for about 77% of the variance. The first axis is strongly correlated with economic prosperity, and the second axis correlated with population density and green space. So based on these metrics, the study cities were shown to be more like their paired city than the other study cities or any other nearby cities. During the winters of 2016 and 2017 and 27 to 2018, data loggers were deployed to record conductivity and pressure readings at 5-minute intervals. This was done in 4 stormwater pipes in each of the 6 cities, so 24 locations. As with the stream data, conductivity readings were used to estimate chloride concentrations, and then pressure was used to estimate water depth. Water depth, pipe geometry, and pipe slope were used to estimate flow volume, and then the flow volume was combined with chloride concentrations to determine a chloride load at each 5-minute time point. Then a chloride yield was generated for each of 9 storm events over the course of these 2 winters. I also identified the drainage area going to each pipe using topographic coverages and proprietary GIS coverages of the stormwater system that were provided by the Metropolitan St. Louis Sewer District. Road area in each of these pipe sheds was then extracted, and chloride yields were evaluated as grams of chloride per square meter of road on a per storm basis. So before I actually present the results, I want to guide you through what you're about to see. On both axes, I'm displaying the yield of salt conveyed during a storm event measured in grams per square meter. On the x-axis is the salt from the city that has brining, while on the y-axis is the salt from the same storm event from the paired city that does not have brine. Every point you see on the figure will represent one pair of cities during one storm event. So any point that falls to the region above the 1-to-1 line represents a pair for which, for that storm, the city that uses only rock salt used more salt than the city that has brine. A point on the 1-to-1 line is a storm for which both cities used the same amount of salt. And a point below the line represents a pair in which the brining city actually ended up using more salt than the city that uses just the rock salt. All right, so as you can see in this results figure, most of the points are in fact above the 1-to-1 line, and there is a significant linear relationship between the paired cities in their salt use. Using a Wilcoxon signed rank test, I was able to determine that this linear relationship is also significantly above the 1-to-1 line. On average, the cities that had brining technology used 45% less salt. So how does this reduction in salt look if we break it down by city? So there's a few notable differences that I want to point out among the cities. But first I want to mention that these cells in gray represent the mean yield across the stormwater pipes within a city for each storm. So those are the means of the 4 different locations in the one city, and then that's for each storm event with the dates listed at the top. This last column is the sum of the means across all of these cities, or across all of these storms, excuse me. So for each pair The city that used brine is the one shown on the bottom in the darker gray. And then positive differences represent instances when the brining city had less salt in their stormwater. All right, so the first thing that I want to point out from this is that our two pairs of more affluent cities shown at the top and bottom of this, this table used more salt than Jennings and Ferguson, which are shown in the middle, and that these two cities used almost the same amount of salt over the course of these 9 storms in 2 years. There are several possible reasons for this. It could be related to the socioeconomic factors. As I said, these two are less affluent cities. But it could also be associated with the microclimate of the area. The intensity of winter storms in this part of St. Louis County tends to be decreased, and at least during our study period it was. And this may be due to the proximity of these cities to the confluence of the Missouri and Mississippi rivers, which may serve as some sort of a thermal buffer for the area. Next, looking at the Manchester-Baldwin pair on the top, these 4 storms shown with an asterisk, those are instances when the city of Baldwin has record of putting down brine. Now, I only had solid records for this for Manchester and Baldwin, not for the other pairs. That's why there's no asterisks in any of the other cities. But yeah, so in those 4 storms, they used brine, and in all instances, they ended up having a decreased amount of salt in their stormwater relative to Manchester. All right, and then looking at the third pair, Webster Groves and Rock Hill, Webster Groves using brine, and in every storm event, they end up using less salt than their paired city. So to recap this third study, my hypothesis was that cities that use brine as a BMP would contribute less chloride to stormwater systems than cities that don't have brine. And this hypothesis was supported in this real-world application of brining in 3 pairs of cities, where brining resulted in about a 45% reduction of chloride. I also found that salt yield varied among cities and among the different storm events, which is unsurprising. The cities are each independently run, and then weather patterns change at even relatively short distances. And since the severity and duration of every storm is different, so too should the response of the public works departments be different. All right, so to summarize the findings of the 3 studies that I've presented this afternoon, we learned that the temperature is an important component of chloride toxicity, and that while cold temperatures can reduce the lethal impacts of chloride, it may exacerbate sublethal impacts. This sort of analysis is imperative if we seek to use the toxicity measures as indicators of pollutant exposures that detracts from environmental quality. And the dichotomy in toxicity versus feeding responses presents challenges in reconciling the short-term lethal impacts of chloride exposure against the potential long-term effects related to the fitness of populations and functioning of ecosystem processes. We have also learned that aquatic macroinvertebrate assemblages respond to chloride pollution not only in terms of absolute chloride concentrations, but also in terms of other characteristics of the exposure profile. Model results suggest that mayfly diversity is significantly reduced as chloride concentration increases. Meanwhile, total taxa richness is reduced when chloride exposures are characterized by higher acute exposures rather than the chronic exposures. And finally, we've learned that the use of brine by city governments has resulted in a 45% average reduction in chloride loads conveyed to streams, demonstrating that brining is a highly viable BMP for local municipal operations. So we've learned a lot about road salt, aquatic invertebrates, and best management practices in the past 5 years. But of course, there's still more to learn. It's well established at this point that most aquatic invertebrates should have a greater tolerance of chloride at colder temperatures. But more detailed study of the impacts of temperature on non-lethal toxicity should be conducted. Most studies of chloride toxicity and toxicity in general have looked at the impacts of concentrations that are stable over time. As we saw in our field study, this is not representative of the types of exposures that we see in streams. So an examination of the toxicity of chloride under variable exposures would be beneficial. In my study of the field response of invertebrate assemblages, taxonomic resolution was limited to order or family for most taxa. I would like to increase the resolution of the invertebrate IDs and run further analysis of the responses to environmental variables. And with regard to the BMP side of the work, municipal governments are among the early adopters of brining technology. There is a need to make BMPs, including brining, more readily available to smaller scale salt users like strip malls, hospitals, and universities, some of which are notorious for over-applying salt on parking lots and sidewalks. Before this technology can be widely recommended, I think we need to have some quality replicated studies on its effectiveness. All right, so now we've reached the end, but there are many organizations and people that I feel I need to thank. First, I want to express my deep gratitude to the EPA Urban Waters Program, Webster Groves Nature Study Society, and PEO organization for their financial support, as Jason mentioned in the beginning. Second, I could not have done the BMP work without the support of the Metropolitan St. Louis Sewer District, and in particular, Roland Beal of MSD, who has been tremendously supportive over the years, as well as the Public Works Department in Jennings, Ferguson, Rock Hill, Webster Groves, Baldwin, and Manchester. And then, of course, I'm grateful to my committee members, especially my mentor, Dr. Jason Knopft, for their guidance and support. And then this work would not have been possible without the assistance both in the lab and in the field from many graduate students, undergraduate students, my family, and then several members of the STREAM team who came out and helped at field sites. And then finally, I want to give a huge thank you to all of the STREAM team volunteers who've contributed data to our winter chloride monitoring program over the past 9 years. Excuse me. These people are the ones who inspired me to start my PhD. They came out and supported me every winter, year after year, to collect and share their data. And they give very generously of their time, both to monitor chloride and other water quality parameters, but also to improve riparian habitat and educate their communities. And these are my heroes. So thank you all for your attention, and I will be glad to put myself together and answer some questions.
Okay, thank you so much, Danielle. All right, I'm going to try something, and as you noticed at the beginning of the seminar, I'm technologically challenged, but I'm still going to try something here. I'm going to click the unmute all button, and possibly we can try and give Danielle a round of applause remotely. So I don't know if this is going to work, but let's give it a shot. So 3, 2, 1, unmute yourself.
Yay!
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 Fantastic. No.
Thank you.
Okay. All right. I just muted everyone again. All right. So yeah, that was great. Thanks so much, Danielle. All right. So I think what we're going to try and do now is take some questions. And so as I mentioned at the beginning, what we'll do is if you have a question, please type it into the Zoom group chat. I will read the question to Danielle, and so everyone can hear it, and also so she can think about the question. And then we'll move on to the next question. And Danielle, I accidentally muted you, so you're going to have to unmute yourself.
All right, I'm back.
Okay, great. Okay, so I will now open it up to questions on the Zoom group chat. Okay, all right, first question is from Nina. And the question is, how do you think saltwater impacts the things that eat the macroinvertebrates?
Okay, so a lot of the invertebrates are eaten by fish, but also birds, frogs, other animals. For the most part, I mean, there's nothing in the salt or in the invertebrates that's going to transfer up the food chain, but there's most fish, and Jason would actually probably have a better answer for this than I would, but it's my understanding that most fish are less impacted by salt than the invertebrates are. So they tend to have a much greater tolerance.
Okay, I do not have a better answer than that. I will just contribute that part. Okay, next question. Benthic invertebrates are more sensitive to other ions. Did you look at the relative contribution of bicarbonate and sulfate?
I did not. Really, within these streams, it was, unless those other ions are coming for some reason with the chloride from road salt, really in these streams, the changes in the conductivity and the changes, which reflected very well in the change of chloride concentration. So I think for the most part, I mean, I guess I don't know, as I didn't actually measure those, but conductivity was very strongly correlated with chloride.
Okay, next question from Steve Blake. What is the relative cost of brine over salting, and how might this influence uptake of this methods? Are there any statistics out there on accident crash rates and economics of infrastructure management that tip the balance economically towards brine?
Basically, brining is one of those miraculous cases where on almost every level, economically as well as biologically, it's better to use brine. There have been several studies showing that it is equally effective, if not more effective, than just putting out rock salt, because what the brine does is it sets up a barrier between the snow and ice and the pavement. And so that means that it doesn't have to melt the snow and ice, it just prevents it from bonding in the first place, and then it melts. So you can use less salt to get the same melting effect. So you can use less, and also this is something I hadn't thought about or heard of until recently, but one of the directors of public works in Webster Groves, he was saying just a couple of months ago that not only that, but they can put brine on all of the streets of Webster Groves in, I think it was something like 8 or 10 hours, and using 2 trucks. But if they want to cover the entire city, they have to have, in rock salt, they have to have something like 5 trucks, and it takes twice as long. So just due to the fact that they're using that much less material, it takes that much less time and effort and probably fuel and all of those other cumulative costs that come along with that.
Okay, next question from Faith Van Drunnen. In your laboratory trials, you found that inverts had greater chloride tolerances at lower temperatures. What implications could this have on winters with higher temperatures or with increased freeze-thaw events? Right.
So the temperatures I was using were around 5 degrees, and the freezing point is at 0. Now, water in streams doesn't tend to get that cold, doesn't go down to 0 very frequently, at least not in the places where the invertebrates are. So that's kind of why I had those temperatures that I used. As far as the implications for freeze-thaw, generally, if there's additional water coming into the stream that is from, let's say, a warm rainfall following after a storm, that additional rainfall would be diluting the amount of chloride. So your concentration of chloride would be going down at the same time that your temperature is increasing. So in theory, I don't envision a whole lot of instances where the water temperature would be warm and you would have higher chloride.
Okay, next question from Sherry Fisher. I'm confused at how you would tease out the impact of impervious surfaces from the chloride use, since the urban areas would also be using more salt.
So, you know, there's a lot of small differences in how salt is used. You know, impervious area includes some things like rooftops that would never receive salt. Impervious surface could include large parking lots that are perhaps not being used frequently. So let's say it's a church parking lot, and if the storm happens on a Monday, they're not going to salt it. So there's a lot of impervious surfaces that may or may not be salted. And so urban development and amount of impervious surface does not always link completely with road surface or with amount of salt used. I mean, there's definitely a relationship, but it's not directly one-to-one. Hope that answers the question.
Yeah, and that surprised me too. I'll say that I thought that it wasn't going to be possible to tease those things apart, but it seems like they're not as correlated as you would think they were. Okay, so we have 2 more questions queued up. Well, make that 3. So let's what I'm going to do is one of them's from Gerardo. I'm going to skip Gerardo's, and maybe we can address that during the exam. And then I'll take the last 2, and then we'll let Danielle get a break. Okay, so next question. Do you think that brining helps keep chloride below the acute and/or chronic toxicity concentrations?
It's difficult to say at this point. I mean, it definitely helps. Anything that we can do to keep chloride off of the road will keep it out of the streams. But I don't know that it's being implemented widely enough yet to see any sort of stream system-wide effects. One of the reasons that I did the study looking in the stormwater pipes is because doing it in stream, you would have too many influences from additional municipalities. It's very few streams end up starting just in one city. And so trying to tease that apart would have been impossible. But until we have a greater number of cities, and then also things like the big box stores and other large parking lot owners, until they start using it as well, it's going to be hard to say that brining has been the solution.
Okay, and then last question from Brian Downs. Are there aspects of your studies, in particular specific results or methodological innovations, that could be generalized to other human applied treatments, for example, road or driveway cleaner or fertilizers?
Right. I mean, in some ways, yes, because anything that we generally do on our landscape is going to end up in a stream. Not 100% of the time, but generally speaking, if you put fertilizer on your lawn, some of that fertilizer is going to go to the stream. If you put a pesticide down, some of that's going to migrate into the stream or into groundwater. And so anything that we can do, whether it's brining or some other best management practice, anything that we can do to decrease our use of chemicals, particularly those that are toxic to aquatic life, is going to be an improvement.
Okay, all right. With that, I think we'll wrap it up and move on to the private exam with the committee. So once again, I really greatly appreciate everybody participating, and I hope you enjoyed Danielle's seminar today. I mean, I know that I've learned just a ton of stuff from her over the past several years, and I greatly appreciate her efforts. So, you know, I think what we'll do is we will have the exam, and then I will try and send out an email to the department with the results of the exam, and maybe you could then get in touch with Danielle and let her know congratulations. So again, thanks so much, everyone. And to the committee,